Assalamualaikum and good afternoon to my lecturer and all judges. My name is Muhamad Hijaz Khairi bin Ma'alwi. My metric number is S47011. So today I will be presenting about my final year project presentation. My title is Isolation and Screening of Fungi Associated with Egg from African Catfish Clarius Garriapinus. And my supervisor is Dr. Nurul Akilah binti Ibrahim. We move to the first introduction. I choose African catfish. It is because it is one of the commercially most important freshwater fishes in Africa, also in Asia, and it is a fish that easier to get anywhere that we want. So here there are a taxonomy of the African catfish and all the biology of African catfish. So this is a total production of catfish species in Asia, and this is a global aquaculture production of African catfish. Move to the common fungus found in fish egg. Fungal disease of fish and fish egg most commonly caused by the Saprolegnia species called water moles. As you can see in the right picture, this is a fungus on untreated channel catfish egg. For the problem statement, fungi are a group of organisms called heterotroph that require living or dead matter for growth and reproduction. The disease that infect fish egg will make production decrease. There are three objectives that I want to highlight here. The first objective, to isolate and identify fungus that affect fish egg, to investigate the virulent characteristic physiological tolerance of isolated fungi, and to observe the pathogenicity of isolated fungus to the catfish egg. For the method, there are three methods that I use in my final year project. The first method is isolation and identifying fungus. There are two ways. The first one is micromorphology and the second one is micromorphology. For macromorphology, we observe with vision, eyes, on the potato dextrose agar, and for micromorphology, we observe under microscope. We put lactophenol cotton blue on the microscope for easier to observe the specific fungus. So the picture here, we can see the type of fungus, the species of fungus. And for the second method is virulent and physiological test, is for to see the production of extracellular enzyme on agar. There are two tests, hemolysine test and protease assay test. And the second one is a physiological test. This is to see the physiological tolerance of the fungi. There are three things that we must I do in the physiological test. For the third method, in vitro egg infection. So here are the method. Move to the result and discussion. I want to highlight the result and discussion because this is the most important for the final year presentation. For the micromorphology structure, agar from film. The macromorphology, we can see here the picture. The picture look like a mushroom look like. And for the spore, the type of spore subsiform, the hyphae, septate hyphae, and the conidiophore. This is conidie and this is conidiophore. For the macromorphology on the agar from skin, we can see here they look like fluffy tuft of cotton-like material, colored white to shade of grey. For the spore, they are periospore, and for hyphae, they are septate hyphae. And the conidiophore, there are four things that important. First one is hyphae here, spore, the small one, and the conidia, and this one is a conidiophore. For agar from gills, they are same like first one. The macromorphology mushroom look like, but the difference is in the spore. The type of spore is globose and oblong, and hyphae is same, septate hyphae, and conidiophore is also same, conidie, and this is conidiophore. For extracellular enzyme types, the first test we do is proteolysis test. This is before, then this is after. The result is no clear zone around the colonies, so the presence of protease enzyme will not happen. And for amylases test, this is before and this is after. The result are positive test result. To make it clear, a clear zone around the line of growth after addition of iodine solution indicates the organism. And the third test for the extracellular enzyme test is lipase test. This is before and this is after. The occurrence of clearance around the medium will be taken as a positive result. For the conclusion, fungi are a rapidly growing organism, but they are not as dangerous as viruses and parasites. Although they are not harmful, fungi are capable of killing small organisms such as fish egg, phytoplankton, and zooplankton. These isolated fungi show virulent characteristics and can cause mortality during egg infection studies. Moreover, the pathogenicity of fungi will be known and what possible treatment prevention can be developed to prevent it. On top of that, this research has not been well completed because the outbreak of the COVID-19 outbreak has restricted the movement of students at the university. However, 50% of the research was successful even though the result did not meet the required criteria. So this is my Gantt chart and this is my reference. Thank you for listening and thank you. Assalamualaikum.